whoever wrote that. Colleagues, welcome. This is the noon meeting being hosted by the Division of Epidemiology and Biostatistics. We are going to start in about 2 or 3 minutes time. So this is just a note to say you're in the right place, particularly if you're online, and if you're in the room, I presume you're in the right place. But we'll give another minute or two for people to filter in because they're still joining in online. And so you're in the right place. We'll start in about 2 minutes. What fraction of this is your PhD?
All of it.
All of it. Okay. Good. The just see. Oh, and great. So we have about 15 or 16 people online and probably another 15 people in the room. So it's a healthy audience for you, Luke. And so welcome everyone to the noon meeting being hosted this week by the Division of Epidemiology and Biostatistics. Our speaker this week is Luke Hannan, who is a lecturer in the division known to many of us. Luke has a background in molecular cell biology from UCT Upper Campus, and then had the privilege of completing our Master of Public Health degree in epidemiology and biostatistics, and has worked in different capacities around the Faculty of Health Sciences as a statistician, working at the interface of biostatistics and omics data. In this case, he's going to be talking about microbiome data, sort of multi-dimensional data sets in biostatistics. And indeed, that's the focus of his PhD, which involves longitudinal analyses of compositional data from, I think it's the nasopharyngeal microbiome, from a cohort here in Cape Town. And we're excited to hear about his PhD. This is kind of the culmination of his PhD work. He's handing in
Very soon.
very soon, not very soon. That's every PhD student's response to when you're handing it very soon. But it really is the end of his PhD process. So this is kind of the fruits and the summation of his PhD project. Luke, we're going to head over to you. We'll go for 30, 35 minutes, have 10 or 15 minutes for question and answer. And we're looking forward to hearing about it.
Thank you very much. So, as Landon mentioned, my name is Luke. I'm a lecturer and PhD student in the division, and today we're going to be talking about mostly my PhD work, which is looking at analyzing longitudinal microbiome data and kind of the challenges associated with longitudinal microbiome data and some of the methods that we use to address those challenges. But before we get into the methods, make sure that we're all on the same page. A little background is probably in order. So the start of my PhD really started with this paper that came out in 2017, which was provocatively titled at the time, Microbiome Datasets Are Compositional, and This Is Not Optional. Prior to this paper in 2017, people typically analyze microbiome data the same way that they analyze many of the other types of omics data, as either continuous variables, like you would in a linear regression, or as count-based variables, like with Poisson regression, those sorts of models. But this paper in 2017 argued that those methods are not appropriate for microbiome data because microbiome data is in fact compositional. Now, what does compositional data mean? or compositional data are any types of data that add up to some arbitrary constant or some arbitrary sum. So I've included the technical definition of compositional data there, but for the purposes of today's talk, that's just for information. We won't really be going through that. You just need to keep in mind that any data that adds up to an arbitrary sum is compositional data. And broadly speaking, there are two main types of compositional data. So you have fixed total compositional data, which are data that add up to a constant sum, and the constant sum is the same for every single person in the study. So classic examples in the health sciences literature are daytime use of physical activity, anything that adds up to 24 hours. Everybody in your study will have, as a sum of those variables, the number 24, regardless of who they are or how their activity differs. Another common example in the health sciences literature is proportions or true binomial outcomes, where your outcome adds up to 100%. Everybody in your study will have that same total sum, 100%. There are lots of literature about analyzing fixed total compositional data. It's a little bit challenging, but the fact that everything adds up to the same arbitrary sum makes things a bit easier from a statistical perspective. Then on the other hand, we have variable total compositional data. And these are compositional data that add up to an arbitrary sum, but that arbitrary sum changes across individuals in your study. And so compositional data as a field really started in the field of geology, where they were looking at analyzing soil samples. And here, the amount of, for example, sand, silt, or clay in your sample depends entirely on how much soil you dug up. But at the end of the day, the total amount of soil that you dug up is a sum of the parts. Similarly, in the health sciences literature, total energy intake is another very common variable compositional type of data, where a person's total energy intake will be variable across the study, if you have different individuals in your study, but that total will always be the sum of the macronutrients. So in this case, protein, fats, and carbohydrates, and it's an arbitrary sum. And then today, particularly my area of interest is microbiome data. And microbiome data is also compositional data, which I'll show you on the next slide, where we see that across individuals, they'll have a different arbitrary total sum, but it's still an arbitrary total sum. So why exactly is microbiome data in general and lung microbiome data in particular compositional? Well, to do this, I've come up with a very brief hypothetical example to explain why it's compositional data. So let's say we have hypothetical sample X, and in this sample, we only have two bacteria A and B, and they are at equal proportions in this sample. If I sequenced this sample on a next generation sequencing machine known as an ion torrent, so the ion torrent has a total predicted read output of between 2 to 3 million reads. So what this means is if you run a microbiome sample on an ion torrent, at the end of the day, the sum of all of those reads that you get out will be between 2 and 3 million. So assuming that I run hypothetical sample X on an ion torrent, I would expect to get between 1.5 million reads for bacteria A and 1.5 million reads for bacteria B. So 50%, 50%. If I take that exact same sample and I sequence it on a different high throughput sequencing machine, for example, the Illumina MiSeq, which is very common next generation sequencing, it has a total predicted read output of between 24 and 30 million reads. And if I sequence that exact same sample, now instead of getting 1.5 million reads per bacteria, I get 15 million reads per bacteria. And so you can see if I were comparing just direct counts between these two runs, even though they're generated by the exact same sample, I'll get very different numbers. And so we're really only interested in the relative abundances of bacteria A versus bacteria B. So why in particular is compositional data analysis a struggle? Not just longitudinally, but in general. Well, it relates back to this problem called the spurious correlation problem, which was first identified way back in 1897 by Carl Pearson of Pearson's correlation coefficient. And he said that when we force data to sum to an arbitrary constant in the compositional data literature, we call that closing the data or a closure. We create artificial negative correlations between the components, even when they're truly independent. So in microbiome data, this is particularly problematic because an increase in one bacteria might appear to cause a decrease in other bacteria, when in reality, it's just an artifact of the relative abundance calculation, or the fact that we forced these bacteria to sum to an arbitrary constant through the use of our next generation sequencing machines. And if you don't believe me, just very briefly, I came up with a simulation to illustrate this point. So here on the top panel, I have 3 bacteria. It's a very small microbiome, but it's simulated. And each of these 3 bacteria are simulated to be completely independent. So when we look at the raw counts in the top panels and the correlation coefficient, you can see that as I've simulated the data, all of these correlation coefficients are functionally 0. There's no relationship between any pairs of these bacteria. But now when I force these bacteria to an arbitrary constant, let's say I sequence them on my sequencing machine, and I rerun the same set of analyses, but now on the compositional data, the relative abundance data, you can see that the correlations go from functionally 0 to strong negative correlations. And this is a massive problem because if you were a biologist trying to interpret this, you might naturally conclude that bacteria 1 and bacteria 2 have a strong negative relationship. And maybe that means something for a disease of interest that you're working on. Or maybe you think that you made a groundbreaking discovery where bacteria 2 and bacteria 1 interact. But it's really just because we forced it to be an arbitrary constant. If we increase the proportion of bacteria 1, by definition, the proportion of bacteria 2 has to go down, because they always have to add up to 100%, regardless of if that's true in the actual host that you sequence your data from. So that's kind of the key challenge around compositional data analysis in general, but also in the microbiome. And my PhD particularly focused on lung microbiome data for 2 reasons. One, as many of you know, lower respiratory tract infections are still one of the leading causes of death in children under 5 globally and in sub-Saharan Africa. And in many cases, it's possible to identify the pathogen that caused the lower respiratory tract infection. And these pathogens are often bacteria that are parts of normal, healthy lung microbiomes. And so the idea there is to look to see if there's some microbial composition or some group of compositions that predispose children to getting lower respiratory tract infection. So that's kind of the public health aspect of my PhD. So just a brief outline of the rest of the talk today, now that we're all kind of on the same page into the background. We're going to be talking about those highlighted bullet points. In order to assess any of the methods that we talk about, particularly in statistics, we need to know the truth. And in microbiome data, that's very hard. So we have to generate a microbiome simulation so we know the truth, so we can evaluate the statistical methods. And then the next 2 parts are kind of using the ALL transform, which we'll get to in a moment, and then compositional functional data analysis. 2 different methods or 2 different approaches to see at analyzing longitudinal compositional data. So before we get onto those methods, as I said, in order to evaluate any statistical methods, we need to know the truth, and we have to simulate microbiome data. Along with compositionality, which I've also already mentioned, microbiome data has 2 other unique challenges. So the first one is sparsity. Depending on the sequencing machine you used and the microbial environment that you're interested in, microbiome data sets can have upwards of 99% zeros. If you take your table, which means it's very challenging to analyze for traditional statistical methods. And the other one is overdispersion. Now, overdispersion in the context of microbiome data typically means that the variance of these microbes is much, much greater than the mean of those microbes in your sample, which is also a challenge for statistical data analysis. And so lots of people have attempted to simulate entire microbiomes with a single parameterization. Unfortunately, it turns out that it's pretty much functionally impossible to simulate an entire microbiome using just a single distribution or a parameterization. So I show that in my PhD, but there are also many other people out there who have shown that you essentially, due to the challenges of microbiome data, can't use a single parameterization. So for the purposes of my PhD and simulating lung microbiome data, what we did is we categorized the microbes in a given microbial environment into one of these 3 categories. So low abundance microbes, medium abundance microbes, and high abundance microbes. And the idea is then we can simulate bacteria from each one of these abundance groups and then combine them all together at the end. Now, typically in lung microbiome data set, as I said, you'd have upwards of 99% zero values. So in my particular simulations, we've set it so that 97% of our microbes will be these low abundance zero value microbes. 2% will be medium abundance, and that abundance level is set to reflect an actual microbiome data set, which we'll talk about in a couple of slides. And 1% will be high abundance microbes. And if we simulate the microbiome data set in this way, it allows us to simulate a single time point. And then once we've generated a single time point, we can apply any method that you would like to generate longitudinal correlation. In my particular PhD, we just used the mean for simulating microbiome data sets in the absence of any change. There's also, we won't talk about it today, but part of my simulation talks about how to simulate microbial change over time. But for many of these methods, we want to analyze their performance on data sets that don't show any change, so we can see how they perform kind of in the base case scenario. So this is what a simulated microbiome data set looks like using the simulation that I've just described. Here we have 100 individuals, although I've just shown the first 20, simulated over 100 time points as well, which for microbiome sequencing studies is a lot. They don't usually follow that many people for that many time points, but because we have simulated data, we can do whatever we want in essence. In order to use this to evaluate the methods that I'll be talking about later today, we need to make sure that it accurately replicates actual microbiome data. So I'll talk about the data that it's replicating in a moment, but here we can see the observed alpha diversity from the actual data set. And alpha diversity, for those of you who aren't familiar, is a measure of intra-individual diversity that is commonly used in the microbiome literature. It basically gives you an idea of how diverse an individual's microbiome is. So this is from a real nasopharyngeal microbiome data set, and this is from our simulated data set. So quite similar, we're on the right page. Then there is another summary statistic that is often used in the microbiome literature, which is beta diversity. So if alpha diversity is intra-individual diversity or variation, beta diversity you can think of as inter-individual variation. It's how different is my microbiome to somebody else's microbiome. And it gives you a sense of kind of the distance between two microbiomes. And here again on the left, we have the observed beta diversity from the real nasopharyngeal microbiome data sets. And on the right, we have simulated beta diversity, which are almost identical in this case, which is promising because it means that the simulation that we've generated can accurately replicate a real longitudinal microbiome dataset from the nasopharyngeal microbiome. So then we can use this method and the actual data to evaluate all of the methods that I'll be talking about later today. Just to give you an idea of what that simulation would look like. Earlier, I showed you the figure of each individual. Here, this figure shows each microbe in our simulated dataset. So each plot is a bacteria, and we can see the abundance of the bacteria over time. In this particular simulation, we've simulated no change, so we should see a flat line across time. And as we can see, we do see, in general, a flat line across time with some variation across time. And then, as I mentioned, all of the simulated data was simulated to try and replicate a real microbiome dataset. That real microbiome dataset is from a study called the Drakenstein Child Health Study, which was a longitudinal birth cohort of 1,143 mothers in Paarl here in the Western Cape. And they followed the mother-child pairs for up to 5 years in life. The data for my particular PhD was from a sub-study of that overall parent study called the Healthy Cohort Study. So in the Healthy Cohort Study, they sampled 103 participants who had never had lower respiratory tract infections. They could have had other illnesses, but the primary inclusion criteria was that they could not have had lower respiratory tract infections. And on those 103 individuals, they sampled the nasopharyngeal microbiomes every month in the first year of life, and then every 6 months after the first year of life. So it's quite an in-depth look at how the microbiomes of these individuals change, particularly in their early years of life. We're not going to go over this, but if those are interested, this is how the actual sequencing was done, kind of the bioinformatics pipeline. If anybody has questions about it, we can come back to this at the end. And to give you an idea of what that data looks like, as I mentioned, lung microbiome data in particular is pretty challenging. And so you can see it's very, very noisy. We have spikes across a number of different microbes. And in particular, in the lung microbiome, it's dominated by a small number of microbes in early life. In particular, you see these blue lines on all of the plots, that's Moraxella. And in this particular data set, almost all of the children are colonized by Moraxella early in life and remain colonized by Moraxella throughout early childhood. And then in the lung microbiome, it tends to stabilize later in childhood. sort of around 4 to 5 years of age, we can kind of see microbiomes stabilizing in each children. So now that we know about the data and the problems with the data, how do we actually analyze microbiome data? So there's a whole field of statistics called compositional data analysis that aims to look at analyzing these compositional data problems. For cross-sectional data, there are a number of methods that have been used in many cases to analyze compositional data, but all of them, broadly speaking, make use of log ratios or log ratio analysis. And as the name suggests, log ratios are just the ratio of two parts, and then we take the log of that ratio.
Sorry,
Yes.
is there a clear correlation between the microbiome that's present in the nasopharyngeal space compared to the lower apices of the lung? Because that's when
Yeah, that's a good question. The idea is that there is some correlation between the nasopharyngeal microbiome and the lower kind of lung microbiome. It's just incredibly difficult to get kind of lung microbiome samples from deep inside the lung. So that's typically why lung microbiome studies, we use the nasopharyngeal microbiome. Yeah, good question. So log ratios are used in compositional data analysis to kind of get around the idea that everything has to sum to 1. In fact, what log ratios do is they take us from the simplex space or the compositional space into the real number space. And so once we've transformed our data via log ratios, we can then analyze our data with any of the classical statistical methods that you're all familiar with. Broadly speaking, there are 3 main types of log ratio transformations. We have the additive log ratio transformation, which is considered the simplest log ratio transform. Essentially, you pick a particular bacteria to act as the reference category, and then all of the ratios of all of the other bacteria are with respect to that reference category that you've chosen. It's not used that often in microbiome data, simply because picking the reference category is very contentious in the literature. We'll see in a moment about some ways that we can go about doing that in a systematic way. The most common approach in microbiome sequencing data is the centered log ratio. The centered log ratio you can think of as the change in one bacteria relative to the average composition of all of the bacteria in your sample. So it's kind of a change from the average. In particular, it's the whatever bacteria that you're interested in as the numerator, and then the geometric mean of all of the bacteria in your sample as the denominator. Very common. And if we have time at the end, we'll go over some of the challenges with all of these log ratio transforms. There is a third one that I feel that I need to mention called the isometric log ratio. It has some very nice mathematical properties. We're not going to talk about it today because it's functionally uninterpretable. And if we have time at the end of today, we can go into why it's uninterpretable. So for today, we're going to be talking mainly about the additive log ratio and the centered log ratio. So the first part of my PhD was asking the question, can we use this simple log ratio, this additive log ratio, to answer complicated microbial dynamics questions? As I said, the ALR is not really used in microbiome analysis, and we wanted to see if it was suitable at all for microbiome data, particularly longitudinal microbiome data. So as I said, this is the equation for the log ratio. It's considered one of the simplest methods because the researcher decides on the reference category, and then you're just comparing everything with respect to the reference category. Now, I'm not the first one to suggest using an ALR transform. There was a paper by Michael Greenacre in 2021 that suggested using the ALR transform for microbiome data, but they really just evaluated its performance on cross-sectional microbiome data. But what they suggest is that you first need to determine if the chosen reference category meets a certain set of criteria. Those criteria are: 1, the reference has to have constant or near constant variance. The idea is that if your reference is not changing, it's much easier to compare across time points and across individuals if your reference remains constant. Ideally, it would be non-zero, simply because having 0 as a denominator in anything makes life very challenging. And in fact, if we have time at the end of today, we can talk about the problems of 0 values in microbiome data in general. And then the third one is that it accurately replicates the original log ratio geometry of the data set. We're not going to talk about the details of that, but suffice to say we compare our transformed data set to our original data set and see how closely they match. And an ideal ALR reference category would mean that the transformed data matches our original data set exactly. And Green et al. In 2021 suggests that although this may seem like an arbitrary process, if we evaluate every possible reference category in our microbiome dataset, we should be able to generate a list of potential candidates and pick from those potential candidates based on these data characteristics that I mentioned and also domain knowledge. So they evaluated this on cross-sectional data, and I thought, well, maybe we could do the same thing for longitudinal microbiome data. And that is what we did. Here, the Procrustes correlation values on these plots are essentially a measure of how well the transformed data set matches the original data set. And ideally, we would want a value of 1, kind of in the ideal world. But in that original paper by Green et al., they suggest correlation values of above 0.95 are generally considered appropriate to be used as an ALR reference category, but the higher the better, along with low and no constant variance. So here on the left, I've used my simulated microbiome dataset from earlier. Here I know the exact characteristics of this dataset, so I can tell what makes this method perform well or not. And you can see in this case, as we increase the variance, as we decrease the variance of our simulated dataset, we can increase the Procrustes correlation, which makes sense given the criteria that we've mentioned earlier. And so based on the simulation, we can see that we can actually use the ALR transform to accurately replicate our data. This was on longitudinal microbiome data. But another question of interest is how does this actually apply to the real world? Simulated data is often the best case. And unfortunately, in the real world, our data doesn't usually follow the best case, particularly, for example, lung microbiome data, with all of its unique challenges. So I applied this method to the healthy cohort data set that we saw earlier. And to be honest, I was not expecting this to work when I first did it, but we can actually see that it's kind of hard to see on this figure. We can see that we generate 2 possible reference characteristics, reference ASVs from that original lung microbiome data set. We have ASV 166, which is a member of the Lactobacillus genus. We have its Procrustes correlation there of 0.95, which is kind of just right at the cutoff for an appropriate ALR. We have relatively low variance. The variance here is log ratio variance, not normal variance. And then we have another possible candidate from the Trachea genus. Again, slightly lower correlation values, below 0.95, so perhaps not as appropriate as a reference characteristic, but also very low variant. And then we would use these and other domain-specific knowledge to pick a reference category. In this case, we know from the literature that lactobacillus is quite prevalent in the lung microbiome of most people, and it's generally considered kind of not really harmful and pretty stable in the lung microbiomes. Leptotrachea, there's not as much literature around it in the microbiome. So in this particular instance, we've decided to use lactobacillus as our reference category going forward. And then once we've ALR transformed our longitudinal data, we can apply any sort of statistical analyses that we would like. So just as an example, I decided to use a hierarchical Bayesian model here with some variable selection. So we have sort of horseshoe priors here to try and penalize our covariates, because in a typical microbiome data set, if you include all of the ASVs in your model, you'll have about 1,000 or so covariates in the model. And here we were comparing microbial composition between HIV unexposed infants and HIV exposed uninfected infants. And again, I wasn't really expecting to see any strong signals given the nature of the data and the small number of people. But in fact, using the ALR transform and the Bayesian model, we draw out 2 ASVs that potentially appear to be associated with infant HIV exposure. One of those ASVs is a bit hard to see, but ASV 150, the second row there, is Streptococcus. And there's a whole host of literature that suggests that Streptococcus is associated with infant HIV exposure. So it's good that we replicated some findings that are already out there. But that first ASV, ASV 171, is a member of the Bacteria genus. I couldn't find any literature on this and its association with lung microbiomes data when I was going through for my PhD. But it appears to be very strongly associated with infant HIV exposure. But in this particular instance, more research is probably needed, but it's an interesting signal that shows that using the ALR to transform can adequately capture the variation in your longitudinal microbiome data sets. So to summarize, we can say that if the characteristics of your data allow it, and they require some careful investigation, the ALR transform can be used to appropriately transform and model longitudinal compositional data. So that's the first part, but I know based on those criteria, there are cases where the ALR transform would be inadequate. The big one is if we expect large shifts in the microbiome across time. So you can think of examples like if we give somebody an intervention like antibiotics that will change the entire microbiome, we know by definition that we won't have a constant reference character or part. So we need some more sophisticated methods to deal with those cases when the ALR transform doesn't work. And one of the proposed ways that we can do that is using functional data analysis. So functional data analysis is when we, instead of treating observations as our unit of measurement, we try and estimate functions on our data, and we use those functions as our units of observation. And so the idea was, can we use slightly more complicated methods to answer these complicated microbiome questions? There was a paper released by Stefanucci and Mazzucco in 2022 that applied compositional functional data analysis methods to cause-specific mortality. This is just to show that they were interested in functional PCA, but we can generalize this a bit more. So in their paper, what that looked like is that we have cause-specific mortality within countries across time. In particular, they were interested in European countries. But one thing that I noticed is that we can kind of map this approach onto microbiome data. The individual causes of death could be our individual bacteria or our individual OTUs. The countries could be our individuals, because we expect causes of death within a country to vary across countries, just like how we expect microbiomes to vary across individuals. And of course, time is just time. We're interested in looking at this over time. So I thought, could we apply these techniques, these functional data analysis techniques, to longitudinal microbiome data? Because they'll allow us to visualize overall mean trends in the composition, and they'll allow us to visualize overall mean trends in specific subgroups that we're interested in. For example, people who received antibiotics and people who didn't receive antibiotics. And importantly, it'll allow us to do this while taking into account both the compositional nature of the data and the longitudinal nature of the data. So does this work for longitudinal microbiome data? It turns out it does work for longitudinal microbiome data. I'm not going to show the simulation results here, but when applied to the simulation results, we were able to accurately characterize our simulated data. And so then we applied the same method used by the authors onto our healthy cohort data set in our lung microbiomes. And we can see, using this method, we're able to summarize the trajectories of our bacteria. Here in this plot, each panel is a particular bacteria in the study, and the dark blue and green lines are the average for those particular bacteria over time in this group of individuals. And we can see, as I mentioned earlier, that in particular in lung microbiome data, it's really dominated by very few ASVs for most of the early life. This is ordered by the abundance of those ASVs, and you can see really Moraxella, which is ASV1, as I mentioned, very common and very high abundance. And the others don't really seem to be playing much of a role in the lung microbiome in these infants. But one of the things that these compositional functional data analysis techniques allow us to do is we can actually generate these same plots after we've clustered the data. So it allows us to cluster the data and then generate these same plots. So here, again, we can see the same plot as before, but instead we've clustered the data. 2 clusters was suggested to be the optimal number of clusters for this data set. And we can plot the average trajectory for those same microbiomes within each of our clusters. So in this plot, it suggests to us that there are in fact 2 different types of early lung microbiomes. One that both of them appear to be dominated early on by Moraxella, but in one group, Moraxella abundance declines over time, And SV3, which is one of the Haemophilus bacteria, appears to increase in that group as Moraxella declines. So this is just to give you an idea of how we can use these compositional functional data techniques to get even more information while respecting the longitudinal nature of our data and the compositional nature of our data. So that's pretty much all we have for the talk today, but just to finally close with, a lot of this stuff was very exploratory. So the next steps are, first one is, this was all in the healthy cohort. Do we see these same results in children that have LRTIs, and are there differences in children that have LRTIs? And then the second point is, can we use the output from our functional data analysis techniques as input to classical regression techniques? So does functional regression work after we've generated these compositional functional data? So those are the next steps post PhD. Thank you very much.
So, Luke, thank you. You were 35 minutes almost on the dot, as promised. And that leaves us with time for digestion and questions and answers. What I do want to say is, can we open up the chat as well? And the reason is, people online, I think it is the case that we cannot hear you. You can, even if you're muted, we resolved with Carmen, or we discovered with Carmen earlier, that we cannot hear you, although you can hear us. So can we ask that you put your question, if you have any, in the chat, and then we can answer them, or Luke can answer them as it comes. So I'm hoping there are some requests for your slides already coming through on the chat, Luke, so that's always a good sign. And so let's open up to questions online and in the room. So our first question comes from Haley, and just so people, Haley, please speak up, and then people online can hear it.
Okay, thank you so much, Luke. That was amazing. In the compositional functional data analysis, where you identified these two clusters, was it a research question of interest to see if the clusters were like they were both present in HIV unexposed and infected and in the HIV exposed? The two different groups.
Yeah, that's a good question. And at this point, we haven't looked into that. This was just exploratory, seeing if the method works or not. There are also other issues around clustering and looking at outcomes. But at this point, we haven't looked at that yet.
Good. Luke, you stand there, and I'll still chair, but just so I can see what questions come in. Chris.
Yes, thank you, Luke. A question on the simulation. You did mention that about 99% on the simulation, it looked like 97. So my question is on the, just to check on the sensitivity analysis side, how were the different things that we considered?
Yeah, that's a good point. So the reason that we picked 97% for the original simulation is because, as I said, typically you'll have 99% values, but this particular simulation was designed to replicate the healthy cohort data set as closely as possible. So those were the values that accurately reproduce the healthy cohort as close as possible. All of these methods will apply regardless of the number of zero values, although that's an interesting point about zero values. We'll get back to if there's another question. So we don't expect that any of these methods will change substantially. We would just change the conclusions based on the number of zero values, but that's a good point to make.
Okay, good. We'll come. So online first, Luke, they're both comments. Chantal, you know well, comments. This is nasopharyngeal rather than lung.
Yes.
Yeah, well pointed out and pointed out that as well. And then Joanne Passmore, I think she's joking, but she says if you're looking for a job, let us know. So, but Joanne, I'm going to consider that a joke. But it is flattering. And so I'm keeping an eye on the chat, and we'll come to Alex. But the request again is to speak up so people online can.
Yeah, fantastic talk, Luke, and sort of, yeah, it's complicated for me. So this is probably like a basic question. But when you presented sort of those two scenarios with the two different techniques, can you kind of explain how technology and the detection levels of the technology interfaces with whether this becomes more of a problem to deal with or less. Does that make sense?
Yeah, sure.
Okay.
Okay, that was round one. Are you ready? Round two, off you go.
Yeah, that these slides, yeah. So the question was around how kind of these sequencing technologies impact the results.
Yeah, because presumably, I'm assuming the technology will just advance the lead output becomes data will be the goals. I'm just curious how that interacts with
Yeah, yeah.
analyzing the data.
That's a good question, and it's actually kind of an open area of research at the moment around the technology, because a lot of what I've talked about assumes that we're always going to be dealing with compositional data. These next generation sequencing tools will always give us proportions based on some total output. Currently, the way that we typically get next generation sequencing data or microbiome data is using these next generation sequences. So it will always be compositional, even if we increase the read count. But there is a lot of active research going on trying to essentially generate true counts from the data. If that happens, it'll kind of be a bit bittersweet for me in that if we're able to generate true counts using these sequencing machines, then basically everything that I talked about today doesn't apply to those data. But it will make a lot of people's lives much easier because then you can use traditional statistical techniques to analyze the data. So as far as I'm aware, I haven't seen any metagenomic sequences that can generate true counts, but if the technology does evolve to that point, then yes, most of what I've said today will apply. Yeah.
I'm going to flip back online. We're going to go Haley and then Luke, you want to make a comment but I have to just go back to the online power.
I'm very curious about the computational power that was required to perform all of this. Like how much of this was you and your laptop and how much of this was supercomputer?
So I didn't use a supercomputer, but I have a relatively powerful computer at home. To give you an example, the simulations, I ran a host of simulated scenarios. So I ran 12 different simulated scenarios, and each simulated scenario I ran 100 times. And it took about 3 weeks to finish all of the simulated scenarios. On the analytics side, it's typically much easier to do just because we're not doing the same thing over and over again. So the ALR transform is probably the most computationally intensive when you're looking at all of the possible reference characteristics. On my laptop, on a typical microbiome dataset, that took about 40 minutes to end up with the correct result. But once you have the candidate ASV or bacteria, doing the actual ALR transform is relatively simple, and the analyses from there, it depends on the analysis that you're using. So in my case, where I use the Bayesian analysis, as many of you are aware, that is much more computationally intensive than sort of traditional statistical analyses. So that one took a bit of time, but if you decided to use like a classic linear regression, it would be as computationally intensive as linear regression.
Yeah. Yeah, Sean, please go ahead.
Oh, sorry, I just wanted to ask, in that case, why didn't you use like the UCT-HPC or something like that?
I didn't use the UCT-HPC because I was able to do it on my computer. Yeah, if I were able to do it on my computer, my desktop PC, then I would have investigated using the HPC. But in this particular case, we were able to do it on a desktop. There would be cases, so the healthy cohort data set is, in terms of microbiome data set, not that big, but you can imagine as you get 4 rows and columns, it might become less and less feasible.
3 weeks
To run the simulations,
Yeah. That's crazy.
Yeah. It took a long time, yeah.
Is any of this during load shedding?
Yes, it was. Yeah, I had to restart.
Playing with fire there.
Yeah. But the HPC probably would have been a good idea, yeah.
Thank you, very incredible talk. Sorry for interjecting earlier. From the 2 clusters that you generated, right? I want to know, because you have essentially classified all these microbiomes into 3 groups, the ones that are low minimum and then I think high abundance. Those two clusters that you showed at the end, which cluster did they belong to?
So those clusters weren't related to kind of the description of those earlier microbiomes. Those clusters were generated completely just based on the healthy cohort data set. So it's hard to classify them as low abundance, medium abundance, or high abundance. But the basically top row of that figure that I showed are all of the high abundance bacteria in the nasopharyngeal microbiome, not the lung microbiome. And then the later rows are much, much lower abundance. In the healthy cohort data set, you very quickly hit that low abundance limit. Yeah.
Good. Here we have some more feedback online, just to say, also comments, but to note, Chantal, who is a microbiologist who has worked on the microbiome in the Drakenstein cohort.
Chantal generated this data.
Yeah. And so she obviously knows this data, and she said it'd be interesting to compare to a prior publication that she's referenced in the chat. I presume they did compositional methods in the prior publication and to reanalyze. So that's a useful comment. Shan, you look like you wanted. Oh no, sorry, Chris.
Yes, look, just two comments. One is on because mostly you visually compared the two, the simulated and of course the observed data. So the question is, other than just the correlation coefficient, do you have other maybe formal methods that you use other than just visually?
Yeah.
And then the follow-up to that one is based on the amount of zeros that you have there, what's your thought around maybe the regression part when it comes to how you depending on the amount of zeros that you have in your data, what's your thought around maybe the regression that you could possibly after PhD?
Two excellent questions. So the first question around, did I use other methods to compare the simulations? So yes, so for all of the 12 simulated scenarios, I compared means, variances using statistical tests, and actual abundances. And then we also looked at, you can test for statistically significant differences in alpha diversity and beta diversity. So whenever I generated a simulated scenario, I compared it to the healthy cohort data set using statistical tests, just to ensure that there wasn't anything kind of strange going on. So that's the first part. This is the second question about zero values is actually a very interesting one in the compositional data analysis world. So if we go back to the slides that show you the formula. Zero values in compositional data analysis are a massive challenge because if you have a zero value in your reference character, your reference SV over here in the additive log ratio transform, you're going to end up attempting to divide by zero. And so you can't actually do the additive log ratio. Similarly, for the centered log ratio, because this is a geometric mean in your denominator, if you have any zero values in the geometric mean, because you're multiplying parts together, if you have any zero values, you're going to end up with a zero value in your denominator for the centered log ratio as well, and you won't be able to do the centered log ratio transform. So there's actually a whole host of literature around how to deal with zero values in compositional data analysis and in microbiome data analysis, which we didn't get into today, but there are kind of a couple of common ways that they attempt to deal with this. The first way is somehow attempting to impute those zero values so that they're non-zero. And there are Bayesian methods that do this. Some people will just add a pseudo count, so they'll pick a suitably small number and they'll just add it to all of the zero values. And there's lots of debate around the appropriate methods to do this or not. So in terms of your question, I think you're aiming towards kind of hurdle models and kind of these negative binomial models that will account for these large amounts of zero values. But in compositional data analysis land, assuming we're doing compositional data analysis, we actually can't use those zero values, so we will typically attempt to transform them in some way. Then, like I said, there's a lot of methods and a lot of literature around what is the appropriate imputation for those zero values. Yeah, good question.
Shan? Yeah, that's good.
So I know almost nothing about microbiome data, and I was wondering what the reads actually are. Is each read a bacterium, or because it kind of explains why why it's necessarily compositional or not.
Yeah, that's a good question. So for all of these next generation sequencing machines, there's some variation in how they work, but basically you start with a sample of DNA, then you break up that sample into little pieces of DNA, and then you sequence all of those little pieces. So the read counts are essentially the number of those little pieces of DNA. And then there's, again, a whole host of literature that, for example, Chantal and others will be a bit more familiar with, but then you try and map those little pieces back to known. So that's what those read counts are. They're essentially little pieces of DNA from the host microbiome.
Okay. We are in the closing stretches. Luke, I wanted to ask a big picture question, which is about sort of statistical practice or biostatistical practice, because so Pearson described this problem conceptually, but actually in a way that's quite obviously applicable here over a century ago. And yet people clearly like compositional methods. And to the point where you said in 2017, somebody said this, we have to be compositional. How does that apparently flawed statistical practice, how does that evolve and how is it propagated in a way that, as you explain it, seems a readily apparent shortcoming?
Yeah, that's a very philosophical question, very good question. A large part of it is that a lot of times, initially, the analysis methods for these types of data are largely driven by the people who generate the data. So, for example, the way that microbiome data was first analyzed shared a lot of history with, for example, analyzing other types of data like RNA-seq data where they used counts. And so the data seemed conceptually similar and people thought that it would be appropriate to use. And so it's mostly because oftentimes the development of the technology to generate the data is quite siloed away from statistical analysis. And then also another broader picture thing is that oftentimes in statistics, we reinvent the same term often because people are just practicing the problem in their specific domain. And it very rarely, those techniques leave those domains. So like another common one is in ecology, they often invent new statistical methods, but those methods aren't often translated outside of ecology, and people are essentially left to figure out their own methods independently. So a lot of it is that there really isn't a lot of exposure across domains that kind of leads this sort of thing to happen.
Yeah, so something can be known in one discipline, but unknown to the other, or discovered in the other.
Yeah, exactly.
And that leads on, if I may, to a follow-up question, which is, how do you change statistical practice? Because particularly when in 2017, someone says, this must be compositional, microbiome data must be compositional. And now in 2021 and now 2025, we have people saying that this makes no sense. How do you think about changing the norm, as it were?
Yeah, that's another big question. I will say, just as a brief aside before I answer that question, that we have to give credit to the scientists in the microbiome field. So that paper came out in 2017, and there was a pretty rapid, pretty large shift in terms of shifting from these count-based methods to compositional methods very quickly. There's another very interesting paper in the microbiome world around rarefaction. That there's some controversy around that we won't go into today, but again, that paper came out and people pretty rapidly shifted to sort of the recommended method. So I think we have to give credit to the microbiome scientists in that sense. In terms of changing statistical practice, that is a much, much harder question. People have been trying for a long time to change statistical practice, not just in the microbiome field, but I'm sure many of you are aware around the controversy of p-values and null hypothesis significance testing. And I think the best that we can do is put the information out there, and then if you're a statistician or you're an analyst on these projects, try your best to push back when you get asked to do something that you think is inappropriate. Oftentimes that's not possible given salaries and stuff like that. But as best as possible, try and steer the conversation to the correct methods. I think is the best that we can do.
Yeah, absolutely.
But it's an unsolved problem.
Wonderful. We're at the end of our, oh, no, we have one more question from Joanne. And Joanne says, others are using normalizing processes like qPCR in parallel with compositional analysis to get a better sense of absolute abundance.
Yes, so that is something that we didn't touch on in this talk, but it's actually an excellent point, is that a lot of this basically assumes that we only know the ratios because of the sequencing technique. There is a whole bunch of literature looking at qPCR or other spike-in methods where you put in a known amount of material. And so if you know the amount of material that you put in, even if these relative abundances, these machines are based on relative abundance, you can attempt to work out the absolute amount using that spike in or that sort of qPCR material where you know exactly how much it is. Because the big problem is at the end of the next generation sequencing pipeline, you don't have an idea of what the original values that you started with. And that's kind of what Joanne is talking about. It's actually there's a new microbiome tool that was released literally a couple of weeks ago called Marslin 3, and they have support for including these sorts of spike in absolute values. So it's definitely an area of active research, which gets around a lot of the problems that I was talking about today. Yeah, excellent comment.
Great. Good. So then we are at the end of our time together. And so we owe Luke a huge thanks. An incredibly clear, incredibly stimulating presentation on what is quite a complicated topic. So thank you for clarifying and providing insight. We look forward to seeing you at the next noon meeting. And please, there are lots of requests. We'll save the chat, lots of requests for your slides, and I'm sure you're willing to share them. And so we can circulate those further. And if you have any further questions or ideas or job opportunities for Luke, please feel free to contact him directly. Okay, thanks everyone for attending online and in the room, and we'll see you soon. Thanks, Luke.